Okay, today's question, we're going to look at a cereal dilution at advanced higher level. Don't quote me on this, but I think this could be either the unused paper in 2020 or the 2021 paper multiple choice. So, all you simply do for this, to make it a lot easier for yourself, is you're going to, as usual, with these data handling questions, you're going to just highlight, take a highlighter, and you're going to just put the context into place, and what it tells you is the figure shows how a biologist used cereal dilution followed by plating to estimate the number of cells in a 100 centimetres cubed stock culture of E. coli. So the thing that jumps out here is it's 100 centimetres cubed. And we look at each of the containers with the liquids in them, okay? And we've got the first one has the 100 centimetre original, 100 centimetres cubed original, E. coli stock culture. Then the next one has got 10 centimetres cubed, made up of the 9.9 originally there and the 0.1 taken from the first test tube. And then the other container, test tube, boiling tube, whatever, has got 10 centimetres cubed, made up of 9.9 centimetres cubed, which was originally in there, and 0.1 centimetres cubed taken from the previous test tube. Finally, what they're going to do is they're going to take 0.1 centimetres cubed from the third and transfer it to the agar plate, incubate it overnight, which is going to allow a colony count. Now, how I do this question, okay, from going to what the question is, how many E. coli cells were present in the original stock culture, how I do this question is, I work my way back, okay, so I'm going to start at the 80, and I'm going to go here, and go here, and then I'm going to go there, okay. So, what we've got to do is a few calculations first. The first calculation is from that 0.1, okay, with the 80 colonies. What we need to work out is how many 0.1 centimetres cubed goes into making up the 10, okay, alongside the 9.9 centimetres cubed. And to do that, what we're going to have to do is we're going to times this by 100, okay? And that gives us our first calculation, okay, 80 times 100. But we'll just put that to the side at this point in time. But we need to get back to the original. So what we're going to do is we're going to make another calculation, which is exactly the same, because we're going to be making the 10 in the third test tube, and we're going to be making it up to 10 in the second test tube, which we took a 0.1 centimetres cubed out. So to do this, what we're going to do is we're going to times it by 100 again, and then we're going to do the same for the final test tube. But remember, this time it's 0.1 centimetres cubed, and you're making it up to 100, not 10. Right, so how many times does 0.1 centimetres cubed go in to make up 100? That means we're going to need to times this by 1,000. Okay, so working back the way from 80, we're going to times 80 by 100. We're going to then times that by 100, and then we're going to times that by 1,000. Okay, so what we've got is 80 times 100 times 100 times 1,000. I mean, you can use a calculator for this, right? But from this, the answer that they're going to give you is 800 million. Okay, so we've got the like a viable count. Okay, we've got a usable count from this. So in the original stock solution, there is 800 million E. coli cells. But when we look at the answers, what we need to do is convert it into scientific notation. So for the second part of this, we've worked out how many cells, okay, as a whole number, but what they're wanting in the answer is they want you to put it into scientific notation. So all I'm going to do for this second part to allow us to get that answer is I'm going to write out that 800 million E. coli cells. And what I'm going to do is I'm going to start to get it into scientific notation. I'm going to start with the point here, and then I'm going to work my way back until I get to the 8.0, which is here, right? So I hope you're still following me. If there's any questions at all, then please just put it in the comments and I can go and try my best to maybe make it more clearer to you. So with this point at the end, to get it from here to here, it's going to go 1, 2, sorry, it's going to go 1, 2, 3, 4, 5, 6, 7, and then 8 to there. Okay, so we've moved that point there to get it into scientific notation, and as we had to move that point 8 times, that's going to give us the final answer of 8.0 times 10 to the 8. So the answer to this question is, how many E. coli cells were present in the original stock culture? The answer is going to be D, 8.0 times 10 to the 8, or as a whole number, that's going to be 800 million E. coli cells present in the original stock culture. Okay, so that's a typical advanced higher serial dilution style question. I think it comes under chapter 1, and it's either E or F for the serial dilution. So it is part of the course. You have to know how to do this skill. Okay, so thanks for listening. If there's anything else I can help you with, just drop me a message and we'll get on to it. Okay, thank you.